Okay, welcome. I know I've already done this, but I haven't got caught up with the video making. So, inheritance and pedigrees. And moving the thing along. What's going on here? No, that didn't work either. What is happening here? There we go. Okay, so this one's all about that idea of inheritance. We talked about complete and incomplete codominance, how the environment and epigenetics have an impact, and of course these two important words are genotype and phenotype. So, genotype, we see the effect of the genotype. We don't see genotypes because they are the alleles that you carry. You get an allele from each parent. For most traits, there are more than 2 alleles available in a species. That's always the case, but for some there are more than 2, and you get one from each parent and blend those together in some strange way. We'll talk about those in a minute. So you can be what we call homozygous. That means your alleles are the same. So you can be homozygous dominant or homozygous recessive. I'll get to what those terms mean in a minute too. Or you can be heterozygous. Your 2 alleles are different. If your 2 alleles are different, you're potentially making the 2 proteins. So an allele will produce a protein. That's actually a really important part of this process. If you've got 2 different alleles, often the dominant allele will always mask the recessive allele if they're both made. Sometimes the making of the dominant allele causes the recessive allele not to be made. So there's a ways of masking out the one that is called recessive. The phenotype is what we actually see. So the one we did in class, of course, is tongue rolling, and poor old Joe and I don't roll our tongues. That's because we both are homozygous recessive. We have 2 copies of the recessive allele, and our tongues simply won't roll. Whereas the rest of you could roll your tongues. So you're either homozygous dominant and have 2 copies of the allele for tongue rolling, or you have 1, so you're heterozygous. But having 1 allows you to roll your tongue. So it's seeing the genotype, if you like. It's seeing the effects of the proteins being made. That's phenotype. So we talk about a number of situations. We talk about complete dominance. So this is where one allele is completely dominant over the other. Tongue rolling is completely dominant over non-tongue rolling. My little example there is using bees. Big B, so uppercase B, big-nosed allele, is dominant to little b, small-nosed allele. Now I just made this up, so I have no idea. But that's that idea. So we put a lowercase letter to denote a recessive trait and a capital letter for a dominant trait. This is a convention. And we use these Punnett squares to try and work out what's been going on. So in this case, mum, I put mum across the top, dad down the side. Mum can make eggs that are little b or little b, so she's homozygous recessive. Dad's homozygous dominant, so you can only make sperm that are big B and big B. And of course, when the sperm and egg meet, they make a new individual. So in this case, all of them will get a big B from dad and little b from mum, and all will be heterozygous. They'll all have big noses like dad, but we don't know if they're dominant or not from that view. We need to realise that mum's what we call a true breeder for little noses, and dad's a true breeder for big noses. This is the things you can work out with Punnett squares and pedigrees are next. Incomplete dominance. This is the one that really confused poor old Gregor Mendel, because he worked out dominance and was quite happy with that. And then he's got his white pea flowers and his red pea flowers, and expected the red to dominate the white, and he'd have only red when he crossed them. So he had true breeding whites and true breeding reds, which he'd bred over generations and knew that that's what they always produced. And then he crossed them, and he got pink. What happened there? So of course any artist will tell you put red and white and you get pink, but he hadn't realised that these proteins were being made by the alleles will also do the same thing. They're blended. So in this case we could use capital letters for both of the traits because they're both dominant. Neither dominates the other. And so to be pink, you're RW. So if mum's red, she's got a red allele and a white allele, and dad's got a red allele and a white allele, you'll get 1 red, 2 pinks, and 1 white. So you can follow these patterns. These ratios are really quite important, which we just discussed. I won't go there again. So the other one, of course, when we talked about this idea of blood type, the ABO blood type is a classic example we use. Oh, sorry. Pause that for a minute. Sorry, hang on, Tom. Sorry. Okay, sorry, chaps. So codominance. Now we talked about this as the classic one we study is ABO blood type. So in this situation, you've got two alleles which are incompletely dominant, so we get that blending. So A and B are both dominant and create this AB type, but they're both dominant over little I, which gives you the O types, these are the genotypes and phenotypes. So to be A blood, which I am, you're either homozygous dominant or heterozygote, but A over I. To be blood type B, you're either homozygous dominant or heterozygous B over I. And then it's the heterozygote AB is a new phenotype, the AB phenotype. And then the O phenotype is the absence of A or B. And A and B put a protein marker on the surface of the cell. So this is an antigen. So there are no antigens on the O blood. So these are, again, this is what affected the protein. Oops, get that thing that changed space again. There we go. So we've always talked about the environmental effects on phenotype because it's not just the genotype. We have this classic old nature versus nurture argument, and of course it's both. You're born with what genes you have, and then there's the impact of environmental conditions, family conditions and things. So, some people without the ability to roll their tongue genetically have managed to train it to, make some sort of shape. So it's training. So, you got some environmental effect there. We all go out, well, most of us go out in the sun and get brown. I've got a fair bit of sun during camp week. Much browner face now, or redder face. And so it changes the phenotype of the face. So it changes that skin tone on the face. Tallness, height, some 380 different genes involved in your height. And these can all be affected by nutritional and exercise developments, conditions at certain points of development, right from in utero right through those early years of into now when you guys are just finishing puberty. And that can depend on whether or not you achieve your genetic potential or not. So this is all that environmental effect. And then a bit of study showed these other effects, which we call epigenetic. And the epigenetic effects really are very much like the idea that Lamarck had for evolution, that you could pass these things on. So these epigenetic changes can occur in my lifetime, and I actually pass them to my children, so that they're it can impact the way they express their genes. So it happens in two ways, and essentially it's about this this segment here. So remember the DNA is huge, 2 meters DNA is all bound up, and the DNA gets wrapped around these histones. These histones are little proteins, and we saw those when we looked at it down the microscope at our strawberry DNA. They're little, almost egg-shaped, clear little bits. Those are the histones that these things wrap around, and we've broken some of them out. So we can methylate. So this methyl group, you can see there's a carbon with 3 oxygens attached to it. That's the methyl group. Attaches itself to cytosine, and by doing that, it causes this gene to become inaccessible. It binds it up. And the same thing happens to these epigenetic factors. Again, there's not methyl groups in this case. There are other things, other chemicals, but they also cause bits of DNA to be bound up and not be able to be expressed. And some things occur naturally. I mean, my grey hair is, the natural process. I'm not requiring to coloured hair anymore. I'm old, so I'm not making that colour. There's also many genes are switched off all over your body. Every one of your cells has all the genes, but they only need to express those genes that relates to their function. So they switch everything else off, and these switching off effects are these epigenetic effects, and we can alter those. So things like, I've got my head in the way there, haven't I? Things like stuff happening in utero and during development. Those environmental chemicals we come across now, because our environment's full of chemicals we've produced. The drugs and pharmaceuticals we take for illness, aging and diet, just quite natural things, can be a source of methyl groups, causing these switching off to occur. And in the end, that can mean cancers and autoimmune diseases, mental disorders, diabetes. One of those mental disorders that I think is probably associated with this is schizophrenia. So we know that intake of THC with cannabis, if there's an ability to become schizophrenic in a family, then that the presence of that drug can cause that. So it may well be an epigenetic factor unlinking a gene or linking a gene, or exposing it or not exposing it. So they can have quite devastating effects. I think that's my last slide. Here we are. So essentially your phenotype is the genetics you have, those genes, the genotype, plus impacts from the environment, plus these epigenetic impacts. And that creates you, your peculiar, particular, different self. So hopefully all that makes sense. And that's genotypes and phenotypes.